So here we're going to get into the next really big thing with this chapter, and that's gas exchange. And so we're going to talk about external and internal gas exchange or respiration. We're going to talk about Dalton and Henry's gas laws, and talk about like how these things happen, what ventilation and perfusion is again. We're going to talk about how oxygen and carbon dioxide are carried in the blood and get a little bit more familiar with hemoglobin. We kind of talked about it a little bit, and then we'll talk about the chloride shift and reverse chloride shift and how they relate to gas exchange, and then talk about oxygen and hemoglobin and the affinity between those two in the lungs as well as in the tissues. All right, so gas exchange, as we mentioned often at this point, we have two sides of this. We have the external side and the internal side. External side is the exchange happening in the lungs. Internal is what's happening between the tissues. And so we have to know these laws to understand how oxygen and carbon dioxide make these exchanges. So we have two laws that we need to know. Dalton's law, which is the law of partial pressures. This basically means that when you have a mixture of gases, like a gas mixture, that the pressure that the mixture exerts is equal to the sum of all of the other individual gases' pressures. So if we look at this equation, we have P, the pressure of the total gases, is equal to the pressure of gas X, the pressure of gas Y, and the pressure of gas Z. So if you add the X, Y, and Z pressures up, you get the pressure of all the gases together. So that's why we call them partial pressures, because it's just the pressure that particular gas in a mixture exerts, because the air that we breathe is a mixture. There's nitrogen, there's oxygen, there's carbon dioxide, there's other various things, right? But it's not just oxygen, it's not just carbon dioxide. And then Henry's law, this is basically looking at the solubility of a gas in a liquid, and that the quantity of the gas that will dissolve in a liquid is proportional to the partial pressure of the gas. Right, but again, this is coming down to solubility. How soluble is this gas in the blood? So Dalton's law, right, again, is just looking at that mixture of gas, right? The pressure it exerts is a sum of all the individual gases' pressures. So this is what's going to help drive the diffusion of oxygen and carbon dioxide in or out of the body, right? So when we're looking at this, like let's say we're looking at this picture here, we see oxygen, this is the alveolus, so this is the air, this is the alveolar air, here's our capillary, so here's our bloodstream, and we see that the partial pressure exerted by oxygen in the air is 100. The partial pressure of oxygen in the bloodstream is 40. Now, just like a lot of other things we've talked about with diffusion, we're going from high to low. And so oxygen is going to go from the high pressure number to the low pressure side. And so that means we're moving from the side with 100, we're moving over into the side with 40, which means oxygen is moving from the air into the blood. On the other side, we have 46 for the carbon dioxide's partial pressure in the blood. It's 40 in the air, and so we see that we're going from 46 to 40, we're moving from the blood out. So then Henry's law comes in. Henry's law, again, is the quantity of the gas that will dissolve. It's proportional to the partial pressure. So we see that as we have more of the gas, we see more of it dissolved in the liquid. This is what's going to lead to things like the bends. This is a scuba diving condition that can occur, especially if someone comes up from a deep pressure, from a deep dive. They're coming up to the surface too fast, then they can get what's called the bends, which is decompression sickness. All right, and this comes down to Henry's law. The other thing that can help is the hyperbaric oxygenation. This is where more oxygen is dissolved into the plasma. We use this to treat people who maybe have they have been deprived of oxygen. We can also use it if someone has like an infected wound, and the wound is infected with bacteria that are anaerobic, because anaerobic bacteria would not want to be in an area where there's a lot of oxygen present. All right, so external respiration. This is really what I care about a lot, right? Is the external respiration, what's going on, and you being able to explain why things are moving the way they're moving. Because I think most of us understand that we inhale, when we inhale air, we're bringing in oxygen and we're getting rid of carbon dioxide. I think most of us understand that point pretty strongly. But the why does it happen? Why does oxygen move in? Why does carbon dioxide move out? That's what I want you to be able to explain. And again, it comes down to partial pressure differences. So in this alveolus, we see that our partial pressure of oxygen is 105, and we look in the blood, we see that the partial pressure of oxygen is only about 40. So in the capillaries, right, we're going to be around that 40, and so we're going from high to low, go from 105 in the air to the 40 in the blood, right? And then as we move away, we see our partial pressure of oxygen has gone up since we've increased the oxygen in the blood. On the other side, we see that we have a partial pressure of carbon dioxide of 46. For the partial pressure of carbon dioxide, we have 40 in the air, and so we're going from high to low, from the 46 in the blood to the 40 in the air. So we see that we've let carbon dioxide escape. So this is why we're moving this way. And there's a couple other things that come into it as well. So the partial pressure differences is one big thing. Also, the surface area for gas exchange, the larger the surface area, the more we can make exchanges. The small diffusion distance, the fact that we're only having to diffuse through 2 cells is big. That's really helpful in this case. And then the solubility or the molecular weight of the gases. Some things are going to move through very easily, right? Other things not so much. So by looking at all these things, when we're looking at the lungs, we see that oxygen is going to enter into the plasma of the blood. Carbon dioxide is going to leave, and this means we go from deoxygenated blood to oxygenated. We're saturated with oxygen, especially bound to hemoglobin. And again, this is what's happening between the alveoli and the pulmonary capillaries. Now, something else that's happening is the ventilation-perfusion coupling. Basically, we are going to make sure that the pulmonary arterioles, the blood vessels leading towards these capillaries where we're going to perform this exchange, they're going to shunt blood to areas where the partial pressure of oxygen, right, is low. We're going to constrict when the alveolar partial pressure is low, and dilate when the partial pressure of the alveolar space is high, right? Basically, we want to increase that blood flow to alveoli when they're full of oxygen and decrease it when it's not, right? So basically what happens, right, is when there's low oxygen, this is going to cause the smooth muscle cells to depolarize, which is going to inhibit the potassium flow. This opens the calcium channels, which then stimulates contraction. Now moving to the internal side. On the internal side, we're looking at oxygen and carbon dioxide. And again, we're going from high to low, right, in terms of partial pressures. In this case, though, the low side for oxygen is going to be in the body cells, in the tissues. So we see that we have, right, a partial pressure in the arteries for oxygen is 100. And as we travel through, oxygen is going to leave because we have about partial pressure 40 of oxygen in the cells, and so we're going to go from 100 to 40. Oxygen enters into the cells. Carbon dioxide is going to be the opposite. We're a partial pressure of 40 for carbon dioxide. It's going to be about 45, 46 in the body cells, and so we're going to move carbon dioxide out into the bloodstream. And so this is how we make that transition. We make that swap between oxygen and carbon dioxide. And this means that the blood, as it leaves these capillaries, is deoxygenated since most of the oxygen has left the bloodstream and has entered into the cells. So there's a lot of other things that can be transported around the blood, but we're really just talking about 2. We're talking about oxygen and carbon dioxide when we're looking at cell respiration. So how do we carry this around in the bloodstream? It's not just free floating, right? Some of it is, about 1.5% of oxygen is going to be just free floating in the plasma, but almost all the other oxygen, 98.5% of it, is going to be carried by hemoglobin, right? So hemoglobin, each hemoglobin molecule can carry 4 oxygen molecules. When there is no oxygen attached to hemoglobin, we call it deoxyhemoglobin. When it does have oxygen bound, it's oxyhemoglobin. And then sometimes carbon dioxide can actually bind to it as well. And this is where we get carboxyl or carboxyhemoglobin or carbaminohemoglobin. So on the carbon dioxide side, about 10% of it's dissolved in the plasma. 30% is going to be carried by proteins, including hemoglobin. And then most of it, right, over half, is going to be transported in the form of bicarbonate ions. And bicarbonate ions are great for this because they help serve as a buffer. And so we can carry this carbon dioxide around without having massive changes in pH for the blood. So we have 3 main mechanisms to do that. Again, some just for the carbon dioxide, some is just dissolved in the plasma, some is carried by the hemoglobin, and then again we have most of it carried by bicarbonate, or in the form of bicarbonate. We have an enzyme that helps get us there. This is called carbonic anhydrase that we find in the red blood cells. When we use carbonic anhydrase, we're going to form carbonic acid, which then dissociates. It's going to kick one of those hydrogen ions or a proton off, right, which then can leave the red blood cell. This also is what causes the acidification of blood. So this is going to decrease the pH. However, we have mechanisms to readily get rid of those hydrogen ions very easily. So that's something we already have mechanisms in place for. So we don't see massive fluctuations in pH. We also have the chloride shift. This is in the tissue capillaries. Basically, those protons in the red blood cells can attach to hemoglobin and attract a chloride ion, right? So we have this chloride ion that kind of binds there, and then we can exchange bicarbonate, right, out of and the chloride into the red blood cells. So this lets us use the plasma to kind of store bicarbonate without changing the charge of the plasma or the red blood cells. And then on the reverse side, right, on the reverse side, in the pulmonary capillaries, we have that increased partial pressure of oxygen, which means we're producing oxyhemoglobin. This means that that proton is going to dissociate from the hemoglobin, and so we end up going back, right? We go back to, we form that proton with the bicarbonate, we form our carboxylic acid, and then we break back down to carbon dioxide and water. And so the chloride can then diffuse out of the red blood cell as the bicarbonate enters in. We flush that from the blood, right? And this helps increase the rate of the gas exchange happening in the lungs. So again, most of our oxygen is going to be carried bound to hemoglobin in these red blood cells. The heme portion has 4 iron atoms, so we have 4 irons per hemoglobin molecule. Each iron can bind to 1 oxygen molecule, and this is where we get the oxyhemoglobin. Oxygen can bind and unbind, right? It can dissociate from it. Things that are going to come into play: the type of hemoglobin, the temperature, the pH, and then how much carbon dioxide and oxygen we have, as well as 2,3-DPG, right? This is basically something that's going to help with controlling how oxygen is released from hemoglobin. Now, normally we have a pretty high oxyhemoglobin saturation percentage, right? We're usually like at 97%. If it's lower than this, we start thinking there's issues with the lungs. We actually, you may have heard, especially with COVID, the stuff going around, or the information going around with people using what's called a pulse oximeter, which is like the little finger clip thing that can tell you, right, what the oxygen, the oxyhemoglobin percentage saturation level is. It's wonderful because it's super cheap, it's non-invasive, it's really fast, it doesn't hurt. Anybody can buy one of these from like a pharmacy, basically. It's something that we can use easily. And there were people who, if they were sick with COVID, were using these to monitor their oxyhemoglobin saturation levels to know whether or not they need to go to a hospital, because it was dropping down too low. Then we know we need to get medical attention right now. So oxyhemoglobin and deoxyhemoglobin absorb different wavelengths of light, and so basically we're just seeing like how much of this is being absorbed at this wavelength of light. And so we can use that to determine how much of the oxyhemoglobin, right, how much of the hemoglobin is saturated with oxygen. So hemoglobin concentration, we have the MCHC, which is the mean corpuscular hemoglobin concentration. Basically, we're looking at the oxygen carrying capacity of blood. With this, it's distinct from the percentage of the hematocrit though, right? So, because hematocrit, that's like how many red blood cells you have. This is where we're looking at the actual capacity to carry oxygen in the bloodstream. If we have less hemoglobin than normal, or because either A, we don't have enough red blood cells, or we don't have enough hemoglobin, we have anemia. And then polycythemia, this is where you have too much. This is usually, we'll see this in like higher altitudes. We also see it in people who smoke. And then, of course, we see it in people who undergo what's called blood doping, which is a method of cheating in endurance sports. EPO, or erythropoietin, is going to be the protein, the hormone that is made by the kidneys to stimulate the production of hemoglobin and stimulate the production of red blood cells in the red bone marrow when we get too low with those oxygen levels. So loading and unloading oxygen from hemoglobin. So when we load it, we're saying hemoglobin is going to bind to oxygen when in the lungs. We go from deoxyhemoglobin to oxyhemoglobin. When we unload, we're going to unbind the oxygen when we get to the tissues. So we go from oxyhemoglobin to deoxyhemoglobin with the oxygen being released. The direction of the reaction depends on the partial pressure of oxygen, as well as hemoglobin's affinity for oxygen. A high partial pressure of oxygen favors loading. A low partial pressure of oxygen favors unloading. Now in the lungs, oxygen has to bind with the hemoglobin, right, so we can move around. So here we need to have a strong affinity for oxygen with the hemoglobin. In the tissues, we need it to drop down, right? We need to lower that affinity so that we can easily let go of that oxygen so that it can be released into the tissues. And so there's a couple things that influence this. The partial pressure, right? A higher partial pressure of oxygen is going to increase the binding for hemoglobin and oxygen. A higher partial pressure of carbon dioxide is going to decrease that. If we're acidic, that's going to decrease the binding for oxygen. So we can unload that oxygen to muscle, because like if muscles are producing like a bunch of carbon dioxide, it's going to be more acidic, and that's going to promote more oxygen being unloaded at those muscles. With temperature, we see an increase in temperature. It means a decrease in binding. So higher temperature promotes more unloading of oxygen. And then the D3-DPG, or the 2,3-DPG, is going to decrease the binding. It's going to just overall lower the affinity for oxygen and hemoglobin. And we have a couple different types. We have HbA, which is just the adult hemoglobin, and HbF, which is the fetal hemoglobin. So normally, hemoglobin has a really high affinity for oxygen, but if it is acidic in the tissues, this changes. So if we're looking over here, we're seeing like the partial pressure and the hemoglobin oxygen saturation. We're seeing as it gets more acidic, we see that affinity drops, right? That affinity is lower than normal. So what we're seeing here, right, when we bind these hydrogen ions to hemoglobin, it's going to lower the affinity for oxygen. It's going to unload oxygen to tissues, and this is going to allow for more hydrogen ions to bind, which then we carry more carbon dioxide in the blood than we were before. Now, red blood cells, they're going to get energy from anaerobic metabolism of glucose because they don't have mitochondria, right? So they can't use cell respiration, which means they're not using up the oxygen that they're carrying, which is fantastic. And so they can make their own 2,3-DPG during glycolysis, but it just takes like a side pathway, a little like side chain, right? Oxyhemoglobin is going to inhibit the production of 2,3-DPG. We're really going to see more of that if someone is anemic or at a very high altitude. And this is going to bind to the hemoglobin. It's going to decrease the affinity for hemoglobin and oxygen, which is going to promote oxygen being unloaded into the tissues. And so we see that it shifts our little curve towards the right. So we see the increase. So here's no 2,3-DPG, right? Bam. And then as we increase the amount from 4.1 to 5 to 8, right, we see that we shift over, right? So we're seeing a lower and lower saturation of hemoglobin. And then different hemoglobins will also have different affinities. The fetal one has a higher affinity than the mom's, so that we can easily transmit that oxygen from, from the placenta to the fetus. When it comes to the 2,3-DPG, this only will work with adult hemoglobin. It will not work with fetal hemoglobin. And then myoglobin, which is really similar to hemoglobin, but it's found in muscles, it only has one heme group. It has a really high affinity for oxygen in comparison. It's going to release that oxygen when the partial pressure of oxygen is low. And this is going to serve as a molecule that can kind of hold on to and store oxygen for us. This is going to kind of be a nice storage and go-between for oxygen in the blood to the mitochondria. And then, of course, different conditions are going to affect the ability for hemoglobin to bind to oxygen, right? So what will cause us to shift left, which means we'll be increasing the affinity, so increasing the binding, is a lower partial pressure of carbon dioxide, a lower hydrogen ion concentration, lower 2,3-DPG, lower temperature, and fetal hemoglobin. Things that shift us to the right, which is a decreased affinity for oxygen. So this is going to let us offload oxygen easier, is going to be a high partial pressure of carbon dioxide, a high hydrogen ion concentration, which means a low pH, higher levels of 2,3-DPG, and a higher temperature. And so if we're just kind of looking, I'm not going to read through this whole thing for you guys. You can pause here to take a look, but this would be a really good summary for you to understand everything we're talking about here. External respiration here on the left, internal respiration here on the right, and kind of what is happening there. You should be able to tell me some of these key things in terms of the direction things are moving, why they're moving in that direction, how things are carried. Those are some key things that you should easily be able to tell me about if I were to ask. Same thing, we're seeing the same thing here. Another summary. Again, you can pause here so that you can take a peek at this. But again, we're just talking about how things are being carried, right? How are we moving things from the alveolar space, right, into the bloodstream? How are we carrying it on blood cells?